Depolarization in the context of biology refers to the sudden change within a cell during which the cell undergoes a dramatic electrical change. Most cells, especially those that compose the tissues of highly organized animals, typically maintain an internal environment that is negatively charged compared to the cell's surrounding environment. This difference in charge is known as the membrane potential of the cell. In the process of depolarization, the negative internal charge of the cell becomes positive for a very brief period of time. This shift from a negative to a positive internal cellular environment allows for the transmission of electrical impulses both within a cell and, in certain instances, between cells. This communicative function of depolarization is essential to the function of many cells, communication between cells, and the overall function of an organism. Physiology. The process of depolarization is entirely dependent upon the intrinsic electrical nature of most cells. When a cell is at rest, the cell maintains what is known as a resting potential. The resting potential generated by nearly all cells results in the interior of the cell having a negative charge compared to the exterior of the cell. In order to maintain this electrical imbalance, microscopic positively and negatively charged particles called ions are transported across the cell's plasma membrane. The transport of the ions across the plasma membrane is accomplished through several different types of transmembrane proteins embedded in the cell's plasma membrane that function as pathways for ions both into and out of the cell, such as ion channels, sodium-potassium pumps, and voltage-gated ion channels. Resting potential equals. The resting potential must be established within a cell before the cell can be depolarized. There are many mechanisms by which a cell can establish a resting potential, however there is a typical pattern of generating this resting potential that many cells follow. The cell utilizes ion channels, ion pumps, and voltage-gated ion channels in order to generate a negative resting potential within the cell. However, the process of generating the resting potential within the cell also creates an environment outside of the cell that favors depolarization. The sodium-potassium pump is largely responsible for the optimization of conditions on both the interior and the exterior of the cell for depolarization. By pumping 3 positively charged sodium ions out of the cell for every 2 positively charged potassium ions pumped into the cell, not only is the resting potential of the cell established, but an unfavorable concentration gradient is created by increasing the concentration of sodium outside of the cell and increasing the concentration of potassium within the cell. Although there is an excessive amount of potassium in the cell and sodium outside of the cell, the generated resting potential keeps the voltage-gated ion channels in the plasma membrane closed, preventing the ions that have been pumped across the plasma membrane from diffusing to an area of lower concentration. Additionally, note that despite the high concentration of positively charged potassium, most cells contain internal components which accumulate to establish a negative inner charge. Depolarization equals. After a cell has established a resting potential, that cell has the capacity to undergo depolarization. During depolarization, the charge within the cell rapidly shifts from negative to positive. In order for this rapid change to take place within the interior of the cell, there are several events that must occur along the plasma membrane of the cell as well. While the sodium-potassium pump continues to work, the voltage-gated ion channels that had been closed while the cell was at resting potential have been opened by an electrical stimulus. As the sodium rushes back into the cell, the positive sodium ions raise the charge inside of the cell from negative to positive. Once the interior of the cell becomes positively charged, depolarization of the cell is complete. Repolarization equals, after a cell has been depolarized, it undergoes one final change in internal charge. Following depolarization, the voltage-gated sodium ion channels that had been open while the cell was undergoing depolarization close again. The increased positive charge within the cell now causes the potassium channels to open. Potassium ions begin to move down the electrochemical gradient. As potassium moves out of the cell, the potential within the cell plummets and approaches its resting potential once more. The sodium-potassium pump works continuously throughout this process. Hyperpolarization, the process of repolarization, causes an overshoot in the potential of the cell. potassium ions continue to move out of the axon so much so that the resting potential is exceeded and the new cell potential becomes more negative than the resting potential. The resting potential is ultimately re-established by the closing of all voltage-gated ion channels and the activity of the sodium-potassium ion pump. Neurons. Depolarization is essential to the functions of many cells in the human body, which is exemplified by the transmission of stimuli both within a neuron and between two neurons. The reception of stimuli, neural integration of that stimuli, and the neuron's response to stimuli all rely upon the ability of neurons to utilize depolarization to transmit stimuli either within a neuron or between neurons. Response to stimulus equals, stimuli to neurons can be a physical, electrical, chemical stimulus, which can either inhibit or excite the neuron being stimulated. An inhibitory stimulus is transmitted to the dendrite of a neuron, causing hyperpolarization of the neuron. The hyperpolarization following an inhibitory stimulus causes a further decrease in voltage within the neuron below the resting potential. By hyperpolarizing a neuron, an inhibitory stimulus results in a greater negative charge that must be overcome in order for depolarization to occur. Excitation stimuli, on the other hand, will increase the voltage in the neuron, which will lead to a neuron that is easier to depolarize than the same neuron in the resting state. Regardless of excitatory or inhibitory, the stimuli travel down the dendrites of a neuron to the cell body for integration. Integration of stimuli equals. Once the stimuli have reached the cell body, the nerve must integrate the various stimuli before the nerve can respond. The stimuli that have traveled down the dendrites converge at the axon hillock, where they are summed to determine the neuronal response. If the sum of the stimuli reaches a certain voltage, known as the threshold potential, depolarization will continue from the axon hillock down the axon. Response equals, the surge of depolarization traveling from the axon hillock to the axon terminal is known as an action potential. Action potentials reach the axon terminal, where the action potential triggers the release of neurotransmitters from the neuron. The neurotransmitters that are released from the axon continue on to stimulate other cells such as other neurons or muscle cells. After an action potential travels down the axon of a neuron, the resting membrane potential of the axon must be restored before another action potential can travel the axon. This is known as the recovery period of the neuron, during which the neuron cannot transmit another action potential. Rod cells of the eye equals, the importance and versatility of depolarization within cells can be seen in the relationship between rod cells in the eye and their associated neurons. When rod cells are in the dark, they are depolarized. In the rod cells, this depolarization is maintained by ion channels that remain open due to the higher voltage of the rod cell in the depolarized state. The ion channels allow calcium and sodium to pass freely into the cell, maintaining the depolarized state. Rod cells in the depolarized state constantly release neurotransmitters which in turn stimulate the nerves associated with rod cells. This cycle is broken when rod cells are exposed to light. The absorption of light by the rod cell causes the channels that had facilitated the entry of sodium and calcium into the rod cell to close. When these channels close, the rod cell produces less neurotransmitter, which is perceived by the brain as light. In the case of rod cells and neurons, depolarization actually prevents a signal from reaching the brain as opposed to stimulating the transmission of the signal. Vascular endothelium. Endothelium is a thin layer of simple squamous epithelial cells that line the interior of both blood and lymph vessels. The endothelium that lines blood vessels is known as vascular endothelium, which is subject to and must withstand the forces of blood flow and blood pressure from the cardiovascular system. In order to withstand these cardiovascular forces, endothelial cells must simultaneously have a structure capable of withstanding the forces of circulation while also maintaining a certain level of plasticity in the strength of their structure. This plasticity in the structural strength of the vascular endothelium is essential to the overall function of the cardiovascular system. Endothelial cells within blood vessels can alter the strength of their structure in order to maintain the vascular tone of the blood vessel they line, prevent vascular rigidity, and even help to regulate blood pressure within the cardiovascular system. Endothelial cells are able to accomplish these feats by utilizing depolarization in order to alter their structural strength. When an endothelial cell undergoes depolarization, the result is a marked decrease in the rigidity and structural strength of the cell by altering the network of fibers that provide these cells with their structural support. Depolarization in vascular endothelium is essential not only to the structural integrity of endothelial cells, but also to the ability of the vascular endothelium to aid in the regulation of vascular tone, prevention of vascular rigidity, and the regulation of blood pressure. Heart. Depolarization occurs in the four chambers of the heart, both atria first, and then both ventricles. The sinoatrial node on the wall of the right atrium initiates depolarization in the right and left atria, causing contraction, which is symbolized by the P wave on an electrocardiogram. The SA node sends the depolarization wave to the atrioventricular node with a delay of about 100 milliseconds to allow the atria to finish contracting. Then causes contraction in both ventricles, seen in the QRS wave. At the same time, the atria are repolarized and relaxed. The ventricles are repolarized and relaxed at the T wave. This process continues regularly, unless there is a problem in the heart. Depolarization blockers. There are drugs, called depolarization blocking agents, that cause prolonged depolarization by opening channels responsible for depolarization and not allowing them to close, preventing repolarization. Examples include the nicotinic agonists suamethonium and acamphonium. References. Further reading: Purvis D., Augustine G. J., Fitzpatrick D. A.L., eds. Neuroscience. Sunderland, Massachusetts, Scheinauer Usuk. ISBN 0-87893-742-0. External links: Depolarization. Psychology Department, Hanover College. Retrieved May 18, 2013.